Good morning, students. It's Kumara here again. In the previous class, we have discussed about some of the aspects of animal architecture. So today, the continued part of the same chapter, particularly with reference to the animal classification, especially the principles of animal classification. So, under which we have already discussed about the basic aspects like outline of kingdom Animalia, binomial nomenclature, international rules of zoological nomenclature, and today let me go through a new topic for you because you have not studied about this in your PU classes, and the topic is numerical taxonomy, which is otherwise called Phanetics, and phylogenetic systematics, which is called Cladistics, and then molecular systematics. First, let me go through Phanetics, which is also otherwise called taximetry. The term phonetics means as observed in the phenotype. You know what is phenotype is? The sum total of external observable characters of an organism is called phenotype. It is an attempt to classify organisms based on similarity, usually in morphology or other observable traits, regardless of their phylogeny or evolutionary relationships. However, phonetics do not distinguish between homologous and analogous organs. Students, you must be aware of the meaning of the term homologous and analogous organs. In general, the phonetic relationship is often summarized with a tree-like network called phenogram. Phenogram is usually constructed by using phonetic data. It contains lines called phenon lines drawn at right angles to the dichotomously branching dendrogram. Phonetic lines represent the percentage similarity of phonetic features between organisms. Let me go through what is dendrogram. It is a diagram that represents relationships among groups of taxa with the highest taxon at the base of a vertical line while the lower taxa branch at appropriate levels. However, some taxonomists have a misconception that phonetics is nothing but numerical taxonomy, but it is not really so. But in fact, most phonetic taxonomy is not numerically based. Generally, phonetics is not characterized by any particular methodology. Let's go through this numerical taxonomy. It is a classification method developed by some taxonomists, but not all, as a part of multivariate analysis. It aims to create a taxonomy using numerical algorithms like cluster analysis rather than using subjective evaluation of their properties. It is based on quantifying observable characters of organisms, so only quantifiable observable characters. In this system, the closeness between groups is determined by the total number of individuals' characteristics which they have in common. Now coming to cladistics, the branch of systematics concerned with inferring phylogeny or evolutionary relationships is known as cladistics. Phylogeny means study of evolutionary relationships between organisms. It is a very common method of classification today. It focuses on the branching of one lineage from another in the course of evolution. Some taxonomists strongly believed that classification should be based on the phylogeny and in particular on the points at which different groups have diverged from each other. Cladistic relationships refer to the paths of the ancestral lineages and thus describes sequence of branching of ancestral lines. Therefore, Cladistics is also known as phylogenetic taxonomy or phylogenetic systematics. Cladistics attempts to identify monophyletic clades or groups. A clade is a group of organisms having single common ancestor and all its descendants. Now let me go through types of clades. First one, monophyletic clade or group. It is a group of organisms consisting of a common ancestor and all its descendants. Thus, a group of species derived from a single common ancestral population is said to have a monophyletic origin. Coming to second type, paraphyletic clade or group. If the group contains its most recent ancestor but does not contain all the descendants of that ancestor, thus paraphyletic group is a monophyletic group only from which one more of the clades is excluded to form a separate group. Now the third type, polyphyletic clade or group. A group that does not contain the most recent ancestor of its members is said to be a polyphyletic group. So let me repeat the thing. Polyphyletic clade, it is a group that does not contain the most recent ancestors of its members is said to be a polyphyletic group. For example, the group consisting of warm-blooded animals is said to be polyphyletic because it contains both mammals and birds, but the most recent ancestor of mammals and birds was cold-blooded. So in this sense, it is referred to as polyphyletic clade or group. Phylogenetics helps us to understand similarity among species or individuals, dissimilarity between species, close relative of a species, whether two groups are identical in evolution, and evolutionary distance between species. So these are the benefits of phylogenetics. Coming to phylogenetic tree. Phylogenetic relationship among various species of a known set is described in the form of a tree called phylogenetic tree. It is nothing but a phenogram that explains the relatedness among the species. It may or may not show the ancestor, that is the root. It is also known as an evolutionary tree or a tree of life because it clearly reveals the phylogenetic relationships or the evolutionary relationships between the groups of living organisms. Phylogenetic tree may be shown as phenogram or cladogram, and in the phenogram the branches are arranged parallel to each other, while in cladogram the branches are radiating outward from central points. So there is a clear difference between a phenogram and a cladogram here. That is what we observed. Both phenogram and cladogram explains the evolutionary origin of various species. Coming to phenogram tree, so the one which you can observe here, you see in the beginning from the common ancestor, three groups or three radiations you can make out, and one will be having about 53% of resemblances, another is having 50, and from that common ancestor these three groups have originated, and from each of this group several subgroups are being originated, as you can see there: LU1, LU2, TL1, PA2, PA1, TL2, etc. So this is referred to as a phenogram. Now coming to the basic differences between cladogram tree and phenogram tree. As you observe clearly here in this diagram, in a cladogram tree, the common ancestor, which is referred to as A here, from which two lines have originated, that is B and C, whereas in the phenogram tree, from the common ancestor, initially there arise two lineages or branches which are referred to as A and B, and from the middle of that another radiation you can observe what is known as or referred to as C here. So this is the clear difference between cladogram tree and a phenogram tree. Structure of phylogenetic tree. A phylogenetic tree is a two-dimensional graphic diagram usually shown as phenogram. It consists of a single root, many nodes and edges. The edges are lines connecting the nodes. They are also known as branches. They denote the descendants and ancestry relationships among different nodes. The node from which all species descended is called root or ancestral node. The root may be placed at the top or on the side. A phylogenetic tree with its root is known as a rooted tree. If the ancestral species or taxon is not known or not considered, it's called a unrooted tree. So if there is no clear ancestor, it is referred to as a unrooted tree. Now, construction of a phylogenetic tree. Phylogenetic trees are constructed using computational phylogenetics method. Distance matrix methods such as neighbor joining method require multiple sequence alignment to calculate genetic distance. Many sequence alignment methods such as Clustal W produce both sequence alignments and phylogenetic trees. Other methods including maximum parsimony and probabilistic interface techniques such as maximum likelihood. So here is the picture which clearly shows the phylogenies which can be depicted having evolutionary relationships. So there are three versions as you can see in this diagram. In version 1, gibbon is the basic or the fundamental organism here from which there arises a lineage which leads to the origin of orangutans, and from which there arises a branch which gives rise to humans, chimps and gorilla. And in version 2, here there is a basal branch from which three simple radiations you can observe. One is leading to human, so the basic ancestor here again is chimp, from which three parallel lines are originating. One leads to human, another leads to gorilla, another leads to gibbon, in between which there arise orangutan. Then in the version 3, here again the ancestral stock will be chimp, from which there arise parallel evolutions like human, one leading to human, another leading to gorilla, another leading to gibbon, and in between orangutans. So you can clearly make out the different versions while construction of phylogenetic tree. Now coming to cladistics, construction of a cladogram. It polarized characteristics like clade. Clade is nothing but the species that share a common ancestor as indicated by the possession of shared derived characters. Clades are evolutionary units and refer to a common ancestor and all descendants. Synapomorphy is nothing but a derived character shared by clade members. Darwin envisioned that all species were descended from a single common ancestor, and he depicted this history of life as a branching tree, which is now called a cladogram. Since fossil records are not complete, scientists rely on other types of evidences to establish the best hypothesis of evolutionary relationships. So here let me further go through what is systematics, the study of evolutionary relationships, and phylogeny, a hypothesis about patterns of relationship among species. Now the further aspects about cladistics, as you observe in this picture, here the basal one, it is referring to the most primitive organism here in this diagram, which is called lamprey, from which there arise the animals or the vertebrates having jaws, then arise the animals having lungs, then arise the animals having amniotic membranes, then you can see a green colored bar is represented here, which represents the origin of animals having hair, and then the animals having lost the tail region, and then finally the animal having bipedal condition, that is walking with two limbs only, that is hind limbs, that is nothing but humans. So this is one good picture which give you a clear idea about what is actually being done in cladistics. So this is another diagram showing some groups of cladistics. So here you can see the first diagram, monophyletic group, and as you observe here in the first phase itself, there arise the giraffes, from which there arise bat, okay, and only bat is being excluded here, hence the group is referred to as monophyletic group. But others, as it is shown in the pink color, those are all having a common ancestor. This is what called paraphyletic group. So in this paraphyletic group, you can see that the pink colored or the pink shaded region, it is with reference to brontosaurs, tyrannosaurs, and other such dinosaurs where they have common characters and they all have the same ancestral stock. And here several parallel lines of evolution can be seen. One is leading to giraffe, another to bat, another to turtle, and another to crocodile. Hence it is known as paraphyletic group. Next comes the polyphyletic group. Here you can clearly make out that bat is having one line of evolution from the basic ancestral stock, and on the other hand it leads to the origin of the evolution of hawk. And in between bat and hawk you can see the presence of turtles, crocodiles, brontosaurs, dinosaurs, and stegosaurs. So these are all the other such uh, animals, particularly the reptiles, which all have the common origin. So except giraffe, the rest of the animals, they have some common features here. So this is another diagrammatic representation of monophyletic group, paraphyletic group, and polyphyletic group. So as you see here in the diagram, in monophyletic group, from the basal ancestral stock, that is A, two parallel lines of evolution can be seen, that is B and C, which they have some common characters, hence they are represented inside a box. And in paraphyletic, A is excluded here, which you can make out very clearly, and B and C have some common characters, hence they are grouped under one box. And in polyphyletic, it is very clearly evident that A and B have some common characteristics, and C is excluded from that group, and D is having some common character, and that leads to several lines of evolution, hence referred to as polyphyletic. Now coming to molecular systematics. Molecular systematics is also called molecular phylogenetics. It is a branch of phylogeny that analyzes genetic, hereditary, molecular differences, predominantly in DNA sequences, to gain information on an organism's evolutionary relationships. From these analyses, it is possible to determine the processes by which diversity among species has been achieved. The result of a molecular phylogenetic analysis is expressed in a phylogenetic tree. Molecular phylogenetics is a broader term that also includes the use of molecular data in taxonomy and biogeography. Molecular phylogenetics makes inferences of the evolutionary relationships that arise due to molecular evolution and results in the construction of a phylogenetic tree. So here you can go through a picture wherein you see that the most fundamental ancestor for all these kinds of animals and plants or the living organisms in general will be monera-like organisms, from which amoeba-like organisms originate, from which cynamoeba-like organisms originate, and that gives rise to two lines of evolution. On one side you see the egg animals have originated, which are referred to as ovillaria, and on the right hand side of that you see the origin of infusoria. So all these are said to be the primitive animals, which are referred to as protozoans, and from uh, planidae there arise gastridae, from which several side branches can be seen, one branch leading to worms, which are called vermis, and another branch leading to plant animals, zoophyta, which leads to some sponges and sea nettles, and then some primitive worms, which are referred to as Ascalminthus, and from which there arise two lineages or branches, one leading to soft worms and another leading to soft animals, that is molluscans, from which there arise a topmost branch, what is known as ringed worms, which is nothing but annelids, and from which arthropods have evolved, and from that arthropod basal branch there arise insects, crustaceans, and star animals. And on the right hand side of that you see the origin of sea squids, that is tunicates, like ascidians and salpae, and these are all said to be the animals which have a common characteristics, that is notochord, hence called chorda animals. And from that there arise the line of evolution leading to the formation of skull-less animals, then jawless animals, then primitive fishes, which are referred to as Selaceae, and from which there arise a right side branch, what is leading to Amphioxus, and on the left hand side you see that it leads to the origin of Myxine, Petromyzon, and some ganoids, and mud fishes have evolved in between, and from those mud fishes protopterans have evolved, and bony fishes called osseous fishes have evolved, like teleosti. So these are all there on the left hand side of that tree, and that leads to the evolution of amphibians, and from amphibians it leads to the origin of a big branch called reptiles, and these reptiles leads to lizards, snakes, crocodiles, tortoises, and one line leads to birds. And from this basal branch of amphibians and a side branch of reptiles, it leads to the origin of primitive mammals, that is promammalia, from which there arise pouched mammals, the pouched animals, semi-apes, apes, ape men, and from ape men there arise chimpanzee, gorilla, orangutans, gibbons, and finally man. So one important thing that you observe here, students, man is said to be in the peak position of this evolutionary tree. So man is regarded as the most highly evolved group of animals in this kingdom Animalia. Then all organisms are composed of one or more cells which will carry out metabolism, transfer energy with ATP, encode hereditary information in DNA. So there is a tremendous diversity of life, like bacteria to whales and whales to sequoia trees, which are the largest trees present on this earth. Biologists group organisms based on shared characteristics and newer molecular sequence data. Here is about some tools of molecular taxonomy. Molecular taxonomy or systematics uses a wide variety of techniques to derive phylogenetic tree. The techniques are the tools like polymerase chain reaction, that is PCR, which is used to investigate variations of DNA on a large scale, and then gene amplification, which is also fundamental to new approaches like DNA fingerprinting, then some genetic markers which are used to make inferences about relationships between environment and morphology, as well as physiology and behavior. And it also incorporates the tools like DNA barcoding and chromosome painting. Now some applications of molecular taxonomy. It helped in clearing up many taxonomical problems. Say for example, Amphiprion Sebe, which had the taxonomic problems like ratified taxonomic status, and that has been resolved by using RAPD technique. 4 species of clownfishes, they had revealed phylogenetic relationships, and that has been resolved by RAPD technique again. Likewise, Indian mackerel populations, a kind of fish populations, they have revealed genetic homogeneity, and they have been resolved by multi-technique approach. Likewise, dolphins, porpoises, whales and dugong, they have developed the capability of accurate identification, and that has been resolved by mtDNA sequences. Now the other applications of molecular taxonomy, it helps in the recognition of the tree of life, which was originally introduced by Carl Woese in 1970s, and he took on an ambitious project to determine the relationships of all life, which took advantage of a molecule that evolves extremely slow, that is rDNA technique, and the DNA that encodes a small subunit of ribosomal RNA. And it is found that sequences cluster in 3 groups corresponding to the eukaryotes. They are Archaea and Eubacteriae. 3 domain classification of the living organisms have finally evolved by using this molecular taxonomy. And applications of molecular taxonomy in the field of medicine make predictions about emerging diseases, and the ability to predict the course of evolution allows scientists to research zoonotic viruses and track epidemic pathogens, and helps in understanding the evolution of pharmaceuticals and drug resistance. And this allows scientists to prepare vaccines for future strains in advance. Now let us go through quickly about advantages and disadvantages of molecular taxonomy. Advantages are using molecules is advantageous for the following 2 reasons. That is, 1, closer to the actual level of heredity. The number of independently varying characters is huge. Each nucleotide is position in theory can be considered a character and assumed independent. The DNA of any given organism has millions to billions of nucleotide positions. Now about disadvantages, homoplasy, especially the reversals, are likely to occur at a higher rate in nucleotide sequences than in morphological characters. And homology among characters is sometimes not easily assessed, and they also have other obvious disadvantages like high cost and intensive training time. And the use of molecular genetics to study the evolution of relationships among individuals and species, to determine the evolutionary relationships of different organisms, which is by the use of nucleotide sequence data, and that is by the uses of molecular taxonomy. So it also involves comparing the sequences of functionally homologous molecules from each organism and to determine the number of differences between them. So greater the differences, more distantly related the organisms are likely to be. So this is all about molecular taxonomy and cladistics and phonetics. So with this, I have completed this first chapter, that is animal architecture and principles of animal classification. So dear students, if you have any kind of doubts regarding this chapter, feel free to post your questions in the group itself. Thank you.